Let's imagine we were living in the 80s and someone told us that computers would soon take care of everything: shopping, appointments, stock market investments, that billions of us would be connected through a network, that we would carry in our hands a device far more powerful than the supercomputers of that time. It would have seemed absurd, but that's exactly what happened. Science fiction became our reality, and we don't even stop to think about it. Now we're at a similar point with genetic engineering. Let's explore its past, present, and an upcoming breakthrough that could forever change how we live and what we consider normal. For thousands of years, humans have practiced genetic engineering through selective breeding, enhancing the most useful traits in plants and animals. We became experts without truly understanding how it worked, until we discovered the code of life DNA, or deoxyribonucleic acid, a complex molecule that guides the growth, development, function, and reproduction of every living being. Information is encoded in the structure of this molecule. 4 paired nucleotides form a code that carries instructions. Modify these instructions and you change the organism that carries them. As soon as we discovered DNA, we wanted to manipulate it. In the 1960s, scientists bombarded plants with radiation to create random genetic mutations, hoping to generate useful varieties. Sometimes it worked. In the 1970s, researchers inserted DNA fragments into bacteria, plants, and animals to modify them, study them, and advance medicine, agriculture, or even just for fun. The first genetically modified animal was born in 1974, making mice a standard model for research that has since saved millions of lives. In the 1980s, genetic engineering became commercialized. The first patent was for a modified microbe that could absorb oil spills. Today, we produce many chemicals using genetic engineering, such as coagulation factors, growth hormones, and insulin, which previously had to be extracted from animal organs. The first genetically modified food hit the market in 1994, the flavosava tomato, which lasted longer thanks to a gene that suppressed an enzyme responsible for decomposition. However, genetically modified foods and their controversies deserve a video of their own. In the 1990s, there was a brief experiment with human genetic engineering to treat infertility. This led to the birth of babies with genetic material from 3 people. The first humans with 3 biological parents. Today, we have super-muscled pigs, fast-growing salmon, featherless chickens, transparent frogs, and glow-in-the-dark zebrafish. All of this is fascinating, but until recently, genetic editing was extraordinarily expensive, complex, and slow. That has changed thanks to a revolutionary new technique, CRISPR. Practically overnight, CRISPR reduced costs by 99%. What used to take a year now takes weeks, and almost anyone with a lab can do it. It's difficult to overstate how revolutionary this technology is. CRISPR could literally change humanity forever. But why did this sudden breakthrough happen? How does CRISPR work? For billions of years, bacteria and viruses have been at war. Viruses, called bacteriophages, attack bacteria by injecting their genetic material, turning them into virus factories. But sometimes bacteria survive and activate their most powerful antivirus system. They store a small piece of the virus's DNA in a genetic archive called CRISPR. If the same virus attacks again, the bacteria create an RNA copy of the archived DNA and load it into a protein weapon called Cas9. Cas9 scans the bacteria's DNA, searching for a 100% match to the virus's sequence. When it finds a match, Cas9 cuts and neutralizes the virus, saving the bacteria. The revolutionary discovery, CRISPR, is programmable. Scientists realized they could give it any DNA sequence they wanted to modify inside a living cell. If all genetic engineering techniques were like using a map, CRISPR is a GPS, precise, cheap, and easy. CRISPR can edit living cells, turn genes on or off, target a specific DNA sequence for study, and work in any cell, microorganisms, plants, animals, or humans. It's a scientific revolution, and it's just the first generation of this technology. Even more precise tools are being developed right now. In 2015, scientists used CRISPR to remove HIV from human cells in a lab, proving it was possible. A year later, they removed HIV from live rats, eliminating 50% of the virus from their bodies with just a simple injection. In the near future, CRISPR therapy could cure HIV as well as other retroviruses like herpes. CRISPR could also help us defeat cancer by enhancing immune cells, making them better at detecting and destroying cancerous cells. All these applications are limited to the individual. The changes die with them, but CRISPR could do much more. Designer babies, editing the human genome at the embryonic stage, permanent genetic changes, altering humanity's gene pool forever. Scientists have already experimented with editing human embryos. In 2015 and 2016, Chinese researchers conducted partial success experiments, showing the challenges ahead, but also that progress is being made. Once the first genetically modified human is born, there will be no turning back. At first, genetic modification will focus on eliminating deadly diseases. As the technology improves, enhancements will become common. Stronger immune systems, better metabolism, perfect eyesight. Eventually, genetic modifications could affect intelligence, physical traits, and even aging. The biggest breakthrough? Ending aging itself. Scientists believe aging is caused by cellular damage accumulation and that genes influence how quickly we age. If we can repair DNA damage and tweak these genes, we might slow or even reverse aging. Some scientists speculate that biological aging could disappear entirely. Death would still exist, but instead of dying at 90 in a hospital, we might live for centuries with our loved ones. This all sounds incredible, but it also raises huge ethical questions. Scene 2. Questions of genetic engineering. Will we create a society that rejects imperfect people? Will genetic selection become mandatory? Could governments use genetic engineering for control or even military purposes? We already pre-select humans based on genetics. Today, many prenatal tests detect genetic disorders like Down syndrome, and over 92% of pregnancies with a Down syndrome diagnosis are terminated in some countries. But CRISPR is not perfect yet. It still makes mistakes. Editing the genome could accidentally activate unknown diseases or cause unpredictable mutations. Genetic engineering is inevitable. It will impact every aspect of life, from curing diseases to redefining what it means to be human. Whether we like it or not, the future is coming. Science fiction is about to become reality. Will we use this power responsibly? Will it help us reach the stars, or will it create new dangers? One thing is certain, we are entering a new era.